Okay, so now we are talking about the chromatin. Chromatin is the final organization of the DNA, which is seen in all stages of the cell cycle. Right? It is seen in all stages of the cell cycle. I'm sure you know the different stages of the cell cycle. What are different stages? G0, G1, M, G2. I'm sorry, G1, S, G2. Please note in M phase we don't see the chromatin. Rather, the chromatin is compacted. Chromatin is compact further to get organized as chromosome. So chromosome will be seen in the M phase. In all other phases of the cell cycle, the DNA will be arranged as chromatin. In all other phases of cell cycle, the DNA will be arranged as chromatin. In the M phase, the compaction occurs and therefore it is the chromosome. Just for example, if this is chromatin, then it becomes more compact. This is chromosome. Alright. So what I want to say is both are same level of organization. Please understand this. In some books and guides it is written that chromatin and chromosome are different level of organization. Chromosome is one level higher. There is no change in level. Only thing is the loose structure becomes more compact so that it can be easily transported. You remember in the M phase the DNA has to be shifted from the equator to the poles. So if it is a loose structure, it will be difficult to transport. Therefore, it becomes the compact structure. The compact structure, but at the same level of organization. It is at the same level of organization. Okay. So let's talk about the organization. First, I'll show you a flowchart of how the different levels of chromatin are there, and then I'll tell you the specific points about each of those levels. Okay. So chromatin can be broadly described to be of two types: the euchromatin and the heterochromatin. Alright, so chromatin can be euchromatin or heterochromatin. We'll talk about each of them later, don't worry. So euchromatin and heterochromatin. The heterochromatin in turn can also be of two types. The heterochromatin in turn can be of two types. We can have the facultative heterochromatin, alright, and we have the constitutive heterochromatin. Facultative and the constitutive heterochromatin. The constitutive heterochromatin based on the banding pattern. Based on the banding pattern, the constitutive heterochromatin can be microsatellites. They can be microsatellites or they can be satellites. We'll talk about it, don't worry. So depending on the banding pattern, they can be microsatellites or they can be satellites. The satellites include two types of structures. These are telomeres and the centromeres. The telomere and the centromere. Okay, so these are different types of the chromatin. Additional points I'll write. The example of facultative heterochromatin is the bar bottle. the microsatellites. Example, triple nucleotide repeats. The microsatellite example, triple nucleotide repeats, they can be 2, 3, 4, 5, 6 nucleotides which are getting repeated again and again. Now let's come to what do you mean by the euchromatin and heterochromatin. So let's quickly see the chromatin and then we'll go to the euchromatin and the heterochromatin. Okay. So chromatin two types, euchromatin and heterochromatin. The heterochromatin further of two types, the facultative type and the constitutive type. The constitutive type can be microsatellites or satellites depending on binding pattern, and the satellites include the telomeres and the centromeres. Alright. So now let us take a look at the DNA. The segment of DNA where transcription takes place, the two strands must be separated. If you don't separate the two strands, the transcription cannot occur. The other segments of DNA will not be separated. Now, if you try to visualize these segments under the light microscope, they will not be visible. Why they will not be visible? Because they absorb the UV light. We cannot see the UV light by the normal eyes. So, what we do to visualize these segments, we'll label them with dye. We put the dye and we will visualize the dye. Wherever the dye is visible, we will assume that the underlying segment is representing the DNA. So let's label them with dyes. We label them with dyes. The dyes will get attached in an equidistant manner. They will get attached in an equidistant manner as per the length of the DNA. As per the length of the DNA, they will get attached in an equidistant manner for both the strands. Correct. For both the strands, they will get attached in an equidistant manner. Right. Now, when you examine them under the microscope, alright, let's say we have one field of vision over here and a similar field of vision over here. I hope they are same size. Let's see. Okay, approximately similar. And when you examine them under the microscope, the DNA will not be visible, only the dye will be visible. We'll extrapolate that wherever the dye is there, it is actually representing the DNA. So let me remove the DNA. Okay, so now we have two fields. Where now the dye is visible. So what you find here, lightly staining. This is the segment which is lightly staining, right? Whereas this is the segment which appears to be darkly staining. Alright, this is the segment where the DNA was uncondensed. The two strands were separated, it was uncondensed. This is the segment where the DNA was condensed, right? This is the segment where the transcription was occurring. This is transcriptionally active. And this is the segment where transcription was not occurring. This is transcriptionally inactive. Alright, so transcriptionally active and transcriptionally inactive, and we already said this is the euchromatin and this is the heterochromatin. So if you get the question for comparison, okay, please refresh if you've not got the link. Okay, okay. Oh, I'm getting complaint that some students haven't got the link for the class. Please message your friends if they have not got the link to refresh their connection. Alright, I hope that will solve the issue for those who have not got the link. Okay. So the euchromatin and heterochromatin, and this commonly comes as the question for write the differences between the euchromatin and heterochromatin. So in addition to the points, you can draw the diagram also, that in the euchromatin the strands are separated and it will be uncondensed, and you can also draw the transcription. You can show the ongoing transcription. I'll draw the diagram here again. I'll draw the diagram here again. So you can draw it like this. This is showing the uncondensed state, and in this you can mark that okay, transcription is also going on. You can label this as the RNA, and you can label this as the DNA. So it covers all the three parts, and you can finally add the dye particles. When you add the dye particles, you need to remove the DNA. You just draw a circle like this, lightly staining, and here you can draw multiple dye particles, and again draw this structure and say darkly staining. So all the 3 points can be shown within the same diagram. I remove the DNA for better understanding, but you can draw the diagram like this, showing the 3 points: uncondensed, lightly staining, and the transcription, the active subject. Whereas on the other side, the condensed, darkly staining, and no RNA is being formed. So very commonly the difference between euchromatin and heterochromatin is asked. This is how we can write the 3 points of differences. Draw the diagram, and finish. If it is only 2.5 marks, that is all you need to do, and you'll get your marks. Okay, let's try to understand the concept of heterochromatin and the euchromatin. We said euchromatin is a segment of DNA which is transcriptionally active, where the RNA is being formed. Now imagine in a particular cell, let's say collagen is formed in the muscle. The collagen is being formed for the extracellular matrix. Is this collagen formed all the time? Similarly, we had talked about the heme. When we talk about heme synthesis, we'll talk about it. We'll talk about the dye later. Don't worry. So when we talk about the heme synthesis, we said there is a feedback inhibition. There is regulation of the heme synthesis when already a lot of heme is there. Then further synthesis of heme does not occur. The enzymes may also not be there. Alright. So please understand that tissue which makes a certain type of protein does not keep making that protein continuously and continuously and continuously. The protein is formed when it is required. The protein itself will have its half-life. Some proteins have half-life running into days and weeks. Some proteins have half-life running into hours. They need to be synthesized more frequently. So the segment which is getting transcribed may not show transcription all the time. It may not show transcription all the time. What this means is the euchromatin is dynamic. Dynamic within the cell. When it is required, the transcription will occur. When the need is not there, transcription will not occur. In addition to being dynamic, it is variable. Variable means in some cells a particular gene is getting transcribed. In the liver, the albumin is formed. The brain does not form albumin. The neuron cells will not form the albumin. The neuron cells will form the enzymes for the neurotransmitters, but the liver will not form the enzymes for neurotransmitters. So the same gene fragment may appear as euchromatin in the neuron, but as heterochromatin in the brain. On the other hand, certain gene fragments may appear as euchromatin in the liver, but may appear as heterochromatin in the neuron. So the euchromatin is dynamic within the cell. It varies depending on the requirement, and it is variable between different cells. Some segments which are euchromatin in one cell may be acting as heterochromatin in the other cell. Okay. Now, if you want to visualize all those segments of the DNA which are getting transcribed, so because of this peculiar nature of the euchromatin, you will have to examine all cells in all stages of the cell cycle and under all stages of development. So we have to take multiple pictures of all the types of cells to check which segments of DNA are euchromatin and which segments of DNA are heterochromatin, right? So even after examining all cells in different phases, some segments are always heterochromatic. What this means, these segments probably do not contain gene which can be transcribed and give rise to any protein, because we have examined them in all the cells in all the different phases and still they are not getting transcribed. We will call such segments as constitutive heterochromatin. We call such segments as constitutive heterochromatin. Right? So those segments of DNA which are never transcribed, are known as the constitutive heterochromatin. Segments of DNA which are never transcribed are known as the constitutive heterochromatin. But this is not so for the bar bodies. What are bar bodies? I'm sure you've come across this term in anatomy. What are bar bodies? When you're doing the histology, embryology, there we have talked about the bar bodies. What are bar bodies? These are inactivated extra X chromosome. See, the term is they are inactivated. Inactivated means they are heterochromatin. They are inactivated means they are heterochromatin. But there is a special condition when they are inactivated. They are extra X chromosome, means until and unless you have more than one X chromosome in the cell, the bar body will not be there. Means if you have a female cell, let's say you have the oocyte and it is undergoing the meiosis, right? So under meiosis, what will happen? The two X chromosome will go into the two different cells. Now, what was bar body where 46XX, that bar body in the meiotic stage where we have the 23X will become the activated form, it will become transcriptionally active. So bar body is inactivated in presence of another X chromosome. So this is the facultative nature. Under special condition, it is heterochromatin. Otherwise, normally, if you separate the special condition, the extra X chromosome if you remove, then it becomes transcriptionally active. So remember, bar body is facultative. It is facultative, means only under special conditions it is heterochromatin. The special condition is another X chromosome. Special condition is another X chromosome, meaning if a cell has, let's say, 46XXX, so in this case, 47XXX, two of them are bar bodies. Alright, if you have the 48XXXX, In that case, the three of them will be the bottom. So only one X chromosome within a cell can be activated. All other X chromosomes are inactivated. All other X chromosomes are inactivated. I don't know whether you have been taught this in your anatomy or not. The one of the X chromosomes has the gene XIST. It produces the RNA. This RNA will coat the other X chromosomes and inactivate them. But RNA has a short half-life, so it must be produced continuously for inactivation. When you separate the two X chromosomes, after some time when the RNA degrades, the X chromosome will become activated. Okay, so this is about the facultative. This is about the facultative heterochromatin. Okay, let's now talk about the constitutive chromatin. We said the three types of the constitutive chromatin are there. We talked about the telomeres. Let's briefly mention what are telomeres. The telomeres are ends of the DNA. They are the ends of DNA. We can examine them in the chromatin structure or the chromosome structure. So they are the ends of DNA. They have a very specific sequence, a repeating sequence which is self-complementary. Repeating self-complementary sequence is seen in the telomeres. They are ends of DNA with a repeating self-complementary sequence. What is their function? What is their function? They stabilize the ends, but most importantly, they protect from DNA repair machinery. Now, this is very interesting. What I've written. They protect from DNA repair machinery. What do you mean by they protect from DNA repair machinery? Why will we want protection from DNA repair machinery? This is very, very interesting. I'll show you the diagram and then it will become clear why we need protection from DNA repair machinery. Okay, let me draw two different DNA. This is end of one of the DNA. You'll notice that I have drawn one end which is shorter than the other end. Now, why this happens, we'll see when we do the replication. We'll not go in detail now. We'll see this when we do the replication. But remember, when you go to the end of DNA, any DNA, one of the strands will always be shorter than the second strand. One of the strands will always be shorter than the second strand. Alright? Now imagine there is another end of DNA nearby. Please note that I have drawn the two DNA particles with different color. I have drawn the two DNA particle with different color, but inside the cell, the DNA particle are not of different color. They are of the same color. Okay, so the cell cannot tell that okay these are two different DNA. For the cell, that two DNA particle will appear same. And now, when the DNA repair machinery comes into picture, and it examines this particular scenario, what will it feel? At first look, what does it appear? Imagine that the two DNA particles are having the similar color, then what is the appearance when you look at this situation? It appears that there is damage in the DNA, there is a break in the DNA. So the DNA repair machinery will start doing its work. What is its work? Its work is the repair of DNA damage. You say, oh, they have got broken, so it will bring the two parts close and it will join them. This is the DNA repair machinery, but now the problem is there. There are other proteins inside the cell. Previously the number was 46. Now, because of the action of DNA repair machinery, the number of DNA particle has become 45. And if you stay in the hostel, you will know that sometimes randomly there can be what is known as roll call. The warden can come suddenly and they can take the roll call. How many are present? How many are bunked away for the night movie? So there can be a roll call. This roll call is done by the local guardian, which is there in the college. We call them the warden. So guardian of genome continuously keeps checking for the integrity of the genome. It keeps checking for the integrity of the genome, and now when it is checking the integrity of the genome, what it finds, only 45 DNA particles are there. There should have been the 46 DNA particles. What will do? It will ring the alarm. It will ring the alarm. The alarm is try to carry out the repair. So look for the person who is missing. If the person is found, very good. But in this case, the person cannot be found because two of them have been joined together to form one. And if the repair cannot be corrected, then cell death. Cell death. So that is why we need protection from DNA repair machinery for the ends of DNA, which is provided by the telomere. Telomere will protect us from the DNA repair machinery. Let's see the organization of the telomere so that you can understand how the DNA repair machinery can be bypassed with the help of telomere. So, like I said, I'm drawing the two strands of DNA with two different colors so that it is easier to understand. Alright, two different colors are being used so that it is easier to understand. Okay, now remember one very important point which we had written previously. And what is that point? Repeating self-complementary sequence. What is the meaning of self-complementary? We'll see. So what happens? The structure that you see here folds upon itself. The structure that you see here folds upon itself. The result is something else. Because of the folding, there is strain at the back end. There is strain at the back end and there is denaturation. In addition, the smaller segment folds and rests over its own end. The smaller segment folds and rests over its own end. The longer segment folds. The longer segment folds, and remember what we had said, it is self-complementary. So now it is able to form the hydrogen bond with itself. It is able to form the hydrogen bond with itself. Alright, it has the hydrogen bond with the original segment also. It has the hydrogen bond with the original segment also, and is able to form the hydrogen bond with itself also because it is self-complementary. This is what I mean by self-complementary. It is able to form the hydrogen bond with itself. It is able to form the hydrogen bond with itself. Another point to note is, now this hydrogen bond which is being formed here, it is as per the Chargaff base pairing. Because it is self-complementary, it is following the Chargaff base pairing. Because of this, the fold will not unfold. It has been stabilized and it will stay there. To increase the stability, there are other types of bonds. Additional bonding occurs, which in this case is known as Hogstein base pairing. This is different from the Chandra base pairing. This is different from the Chandra base pairing, and it is known as the Hogstein base pairing. It doesn't follow the same pattern, but it further stabilizes the folding. It further stabilizes the folding. Now imagine we have another we have another end of DNA, another end of DNA. They have come together. Now, is the DNA repair mechanism able to join them together? No. This is the primary role of the telomere. It stabilizes the end of DNA and protects from DNA repair machinery. If the telomere is exhausted, this protection which is provided will be lost. And if this protection is lost, then we'll see what will happen. The apoptosis will occur. That is why the telomere is important. When we do the replication, then we will also see how this telomere can be lost and how it can cause the apoptosis. About telomere, one more thing I want to tell you. The structure that you see, it is called the D loop. Resembles the letter D, and therefore it is called the D loop. D loop is a feature of the telomere. Okay. Now here, what do you see? In this particular part, what we find, the triple stranded DNA, which is another feature of the telomere. So telomere has the D loop, and it shows a segment where we have the triple stranded DNA. We have the triple stranded DNA in this particular segment. So these are two important properties of the shape of the telomere. When the telomere is organized, it presents a D loop and it shows the triple stranded DNA. This is made possible due to the self-complementary sequence of the DNA. Self-complementary sequence of DNA which is present in the telomere. Let's go through the telomere again very quickly. Very common short note. Why the telomere is important will become apparent when we do the replication. Okay. So telomere are end of DNA. They have the very important property of repeating self-complementary sequence. With the help of this repeating self-complementary sequence, they stabilize the end of DNA and they protect from DNA repair machinery by folding upon itself. Protect from DNA repair machinery by folding upon itself. Right? By folding upon itself, it forms a closed structure which bypasses the DNA repair machinery. This telomere structure will show the D loop, and with the help of Hogstein base pairing, it presents with the triple stranded DNA, which does not allow the unfolding to occur easily. It does not allow the unfolding to occur very easily. Okay, so that was about the telomere. There was one question, which is the dye which is used. Various types of dyes can be used for staining of DNA. Dyes for staining DNA. There are various types of dyes which can be used for staining the DNA. Traditionally, the dye to be used was ETBI, ethidium bromide. But ethyl bromide has been found to be carcinogenic. So nowadays it is being avoided, although it is very cheap and it has limited carcinogenic potential. So it is still being used very widely. However, wherever the resources are not a crunch, we are moving from ATBR, the more common SYBR green, particularly for the DNA. We're talking about the DNA. From ETBR, we move to the SYBR green. The various other dyes which may be used are cytonine, LC green, EVA green. Alright, so these are the newer dyes which are being used. An additional advantage of using these newer dyes, they will only attach to the double stranded. Alright, they will only attach to the double stranded DNA. And fluoresce. Normally they are piston. When they attach to double stranded DNA, only then they will DNA. Then they will fluoresce. Attached to DNA and then fluoresce. Okay. Whereas ETBR is a non-specific dye, so you have to wash it properly before you take the reading. Okay. So these are the different dyes which are being used. Most commonly used was the ETBR, but now we are shifting. Now we are shifting to SYBR. Previously it was the ethylene bromide, but now we are shifting to the SYBR. Okay, so this was the telomere. Let's talk very briefly about the centromere. Although centromere is not asked so frequently, very rarely you get the question on centromeres in biochemistry. You can get in anatomy obviously, but in biochemistry rarely you get the question. From the biochemical aspect, from the arrangement of DNA point, you should know it is the so-called center of DNA. I say the so-called center of DNA because it need not be present at the center. If you recall, it can be, the DNA can be metacentric, it can be sub-metacentric, it can be acrocentric, or it can be telocentric, describing the position of the centromere. Meta means at the center, sub-meta slightly away from the center, acro means towards the end, and telo means right at that. The centromere can be present anywhere. However, it is still called the center of DNA. Although these are repeating sequences, but they do not have a specific pattern. Repeating sequences, but they do not have a specific additional property. However, the function, they have a well defined function. They provide what is known as docking site for microtubules during cell division. Now this should be obvious. When the cell division occurs, the DNA is arranged in the equator and it is pulled to the poles by the microtubules. But these microtubules must attach at some point. They must attach at some point on the DNA. So that point is the centromere. So the docking site or the point of attachment for microtubules. Alright, so only 2-3 points from the biochemical aspect. The centromeres, they are repeating sequence, but the primary function is provide docking site. However, what you have to note, both telomere and centromere have no genes. We already said they are constitutively heterochromatin, and in addition they have no regulatory function. In addition, they have no regulatory function. Both telomere and centromere, they do not have genes, they have no regulatory function. Therefore, they are generally not associated with inherited genetic disorders. They are not associated with inherited genetic disorders. Right? Both telomere and centromere do not have genes, and they are not associated with inherited genetic disorders. Last, we come to the microsatellites. Alright, microsatellites are repeating segments of 2,6 nucleotides. Repeating segment of 2,6 nucleotides. What this means is we have either 2 to 6 nucleotides which are repeating n number of times. N can be variable, it can be 10, 20, 50, 100, 200, can go even up to 1000. So repeating segment of 2 to 6 nucleotide, that is the microsatellite. So I'll try to explain why some segments are called satellite and some segments are called microsatellite. See, when you want to visualize the DNA, I already told you we have to stain it. Alright, when we stain it, we can get different types of bands. A pattern is seen. Now remember, the microsatellites, and we've seen the satellites also, they have repeating sequences, which is not generally seen in the sequence of DNA. Because they are repeating sequence, the banding pattern for these segments is different. A very distinct, different banding pattern. A very distinct and different banding pattern, and very distinct and different banding pattern. If the segment which is showing a different banding pattern from the rest of the DNA is a thick segment, it is satellite. Alright, but if it is a very thin segment, it is called the microsatellite. Alright, whereas these segments are satellites. These segments are satellites. Okay, I hope this clarifies why some segments are called satellites and some are called microsatellites. I told you it depends on the banding pattern. On the banding pattern, when you want to visualize the DNA, based on banding pattern, they are called satellites and microsatellites. Now, the microsatellites are very, very important. They have a regulatory function. Remember, they are constitutive heterochromatin, so they do not have genes, but they have regulatory function because they allow attachment of regulatory proteins which can exert effect both downstream and upstream. These regulatory proteins can exert an effect both downstream and upstream. Imagine if the size where these regulatory proteins are going to be attached is either decreased or it is increased. So it will alter how many proteins are getting attached. If you decrease the size, regulatory protein which are getting attached will be less. If you increase the size, the regulatory protein which are getting attached will become more. So the regulatory function will be modified, and that is why the microsatellites are found to be associated with inherited genetic disorders. This happens when the size of the microsatellites either increases or decreases. Okay, the segment which is getting repeated, if it is duplicated or triplicated or a part of it is removed, that is when they will cause the disease. So microsatellites can be associated with inherited genetic disorders. Obviously, we'll read about them, but later next year in pathology, we will read about them. A group of disorders called the triple nucleotide repeat disorders. Various well-known inherited disorders are due to the triple nucleotide repeat expansion, increase in the size of the triple nucleotide repeat. Okay. So this is about the structure of the chromatin. This is about the structure of the chromatin, and that is the nuclear DNA is completed. We have completed discussion on the nuclear DNA. Let us quickly move on to the discussion on the mitochondrial DNA. Let us quickly talk about the mitochondrial DNA. Any questions from nuclear DNA? Any questions from nuclear DNA? Because now we are moving to the mitochondrial DNA. Any questions from properties of nuclear DNA? Because now we are moving on to the mitochondrial DNA. Now this mitochondrial DNA is a very common, it is a very common question in it is a very common question in your exam. Very frequently you get to write a short note on the mitochondrial DNA. When we get the long question on the DNA, then also the mitochondrial DNA can be added as a small part of that long question, where you may need to write for the mitochondrial DNA for maybe 2 or 3 marks. So we should know some important points about the mitochondrial DNA. DNA repair mechanism will come later. Don't worry, we'll come to the DNA repair mechanism. Okay, that will do once we do the replication. Okay, let's not jump the gun. First we're doing the properties. Okay, so we'll do that once we do the replication. Okay, so let's talk about the mitochondrial DNA. The mitochondrial DNA is double stranded, just like the nuclear DNA, just like the nuclear DNA, but it is not linear. It is circular. It is not linear. It is circular. So it is double stranded but circular. In addition to this, it is having a small size in comparison to nuclear DNA, which runs in millions of base pairs. The size of the mitochondrial DNA is 16569 base pairs. The size of the mitochondrial DNA is 16569 base pairs, which is very, very small in comparison to the nuclear DNA, which is always greater than at least 1 million base pairs. Minimum size is 1 billion base pairs. It can run even up to 100 million base pairs. Chromosome number 1 can have 100 million base pairs. Okay. Now, within these 16569 base pairs, we find 37 genes. 37 genes, only 37 genes are present. Out of these 37 genes, 13 will code for proteins, 2 will code for ribosomal RNA, and the remaining 22 will code for the transfer RNA. Alright, so 37 genes, out of which 13 is for proteins, 2 code for ribosomal RNA, the remaining 22 will code for the transfer RNA. Alright, please note that all the proteins which are coded by the mitochondrial DNA, all the proteins which are coded by the mitochondrial DNA belong to the electron transport chain. All the proteins belong to electron transport chain. If you recall, we had mentioned very briefly that electron transport chain has 67 protein subunits, out of which only 13 will come from the mitochondrial. This means less than 20% of ETC proteins are coming from the mitochondrial DNA. Alright, so less than 20% of the electron transport chain proteins are coming from the mitochondrial DNA. In addition to this very important point, the mitochondrial DNA does not have introns. Now, this is very important. Try to understand what is the implication of not having introns in the mitochondria. Now, in the mitochondria, the nucleus is not there. This is the structure of the mitochondria. This is the structure of the mitochondria, and here suppose the DNA is there. From this DNA, when you make the RNA, the synthesis of RNA is occurring in the same compartment where finally the formation of protein is going to occur. Alright. Now, because intron is not there, we don't need to modify the RNA after transcription. So what happens? This is the DNA. On this DNA, the transcription is going on, and so you get the segment of RNA. Let's say here the synthesis is going on. This part is already synthesized. So what happened? At the same time, the ribosome will come and get attached, and the simultaneous protein synthesis can occur. So because no introns are there, simultaneous transcription and translation, simultaneous transcription and translation occurs, because the RNA doesn't need to undergo modification. The RNA doesn't need to undergo modification. There are very few untranslated segments. There are very few untranslated segments. In comparison, if you look at the nuclear DNA, barely 1.5% is translated. Barely 1.5% is translated if you look at the nuclear DNA. Also, rate of mutation of mitochondrial DNA is high, about 5 to 10 times greater, 5 to 10 times greater than nuclear DNA. 5 to 10 times greater than nuclear DNA. Lastly, when you look at the translation, we call it the genetic code, we find that there are some differences from the universal genetic code. There are some differences from the universal genetic code during that translation. What are those differences? Let me tell you. So what I do here, I'll write the sequence of nucleotide, which is called the codon. The normal or the nuclear translation, and you compare it with the mitochondrial translation. Right. So first codon in this list is UG. Right. UGA is normally a stop codon. We know this. It is normally a stop codon, but in mitochondria, it codes for tryptophan. Alright, in mitochondria, it codes for tryptophan. Then we have AGA and AGG. Normally, they code for arginine. Normally, they code for arginine. But in mitochondria, they work as stop codon. In mitochondria, they work as stop codon. AUG. Normally, it translates as methionine. It is the only codon for methionine, but in mitochondria, it codes for the starting amino acid, which is the formyl methionine. Which is the formyl methionine. So we must have another codon for methionine, AUA, which normally translates as isoleucine, is coded as methionine inside the mitochondria. These are the differences from the universal genetic code as seen in the mitochondria. When you're writing the short note on mitochondria, make sure you mention these differences from the universal genetic code. They are very, very important. Before we go further, I would like to draw a comparison for the mitochondria, which is easy to understand. Now, see what happened during the primordial ocean when it was there. The primordial soup, you know about evolution, how the new cells were formed. So when the evolution was taking place in the primordial soup, the different unicellular organisms had started evolving in the very, very beginning. So the larger cell will eat the smaller cell will eat the minerals and amino acids which are floating in the primordial soup. So one fine morning, the eukaryotic cell engulfed a prokaryotic cell. Alright. So the prokaryotic cell was engulfed. Now it felt okay. I'm going to die. What to do? What to do? So when the eukaryotic cell started digesting the prokaryotic cell, the prokaryotic cell says, "Okay, don't kill me. Don't kill me." So the eukaryotic cell says, "If I don't kill you, what will I eat?" So the prokaryotic cell looked to the left, then it looked to the right, looked up, and it looked down. And he told the eukaryotic cell, "Okay, give me 2 minutes. Give me 2 minutes, and after that, if you want, you can kill me." So what happened? As long as this mitochondria was not there, when the eukaryotic cell does the metabolism, it will be anaerobic glycolysis. At the end of glycolysis, either the lactate or the pyruvate will be there. Now, when the mitochondria comes in, it has the ability to metabolize the pyruvate. So it will take in the pyruvate, which is left behind at the end of anaerobic glycolysis, and use that pyruvate, completely metabolize the pyruvate, and for every pyruvate it will give the eukaryotic cell 12.5 ATP. So normally from a glucose molecule, the eukaryotic cell before mitochondria gets 2 ATP. But when the mitochondria comes into the picture, the same glucose molecule is able to give us, we have seen this, 32 ATP. So there is a net gain of 30 ATP, 15 times or 1500% increase in the ATP production. The eukaryote was very surprised. Oh, this is magic. Okay, I'll not kill you, but you'll have to work for me. I'll not leave you also. I'll not kill you, but I'll not leave you also. You'll have to work for me. In turn, I'll protect you. I'll give you all the things. I'll give you everything you need to work, but you will have to work for me. So the productive cells, oh, very good. So this is the symbiotic relationship. Alright, it is a symbiotic relationship between a primordial prokaryote and the eukaryote. This prokaryote evolved as the mitochondria. That is why when we look at the genome of the mitochondria, we find bare essential genes which are not present in the eukaryote during evolution. Only those are retained. All other genes have been lost, which were having the duplicate of the eukaryote. All other genes have been lost, which are synthesized by the nuclear DNA and given to the mitochondria. The mitochondrial genome contains only those genes which cannot or which are not present on the eukaryotic or the nuclear DNA. Why I am telling this? Remember, whatever we have discussed for the mitochondria is applicable for the prokaryote, in other words, the bacteria. Alright, so all the properties that we discussed for the mitochondria is applicable for the prokaryote and vice versa. What is applicable for the prokaryote or the bacteria will be applicable for the mitochondria. I hope this correlation is clear to everyone. The mitochondria is the primordial prokaryote and it has the similar features to the prokaryotes when it comes to the metabolic functions. Why I am telling you this is now we come to the next question. Increase or multiplication of mitochondria. From where will the new mitochondria come? The very important question. From where will the new mitochondria come? See, when the cell increases the metabolic activity, it requires more mitochondria. When the cell is about to divide, during division, each cell will have only half of the mitochondria. So now the number of mitochondria have to be increased. So there are various conditions where we will need more of the mitochondria. For example, if the sperm cell is getting matured, it is having an army of mitochondria to provide the energy for the wavy motion of the tail, which pushes the sperm cell forward. So there are various conditions where we require a large number of mitochondria. So how will the new mitochondria be formed? Various scenarios are there. Use existing mitochondria as template. Can you do this? Use the existing mitochondria as template and then synthesize. You see that and make more and more mitochondria. Can we do that? Can we use the existing mitochondria as template and make more mitochondria? Unfortunately, no. That is now not how the mitochondria is formed. Then what do we do? Can we borrow from surrounding cells? Some cells do not require a lot of the mitochondria. For example, we have the myocytes around which there may be adipocytes. Adipocytes do not require a lot of mitochondria. So can you borrow the mitochondria from adipocytes for the myocytes? Can we do that? Can we do that? Again, the answer is no. So how will the number of mitochondria increase? How will the number of mitochondria increase? Please give your answers. How will the number of mitochondria increase? How to increase number of mitochondria? What process will be used? How to increase the number of mitochondria? What process is to be used for increasing the number of mitochondria? Anyone? Anyone? I already given you the hint. I just told you a long story. I just told you a long story, which is the hint. How to increase the number of mitochondria? How to increase the number of mitochondria? Anyone? The answer is the answer is just like prokaryotes, means binary fission. 1 into 2, 2 into 4, 4 into 8, 8 into 16, like that. Just like prokaryote, it will grow in number by binary fission. So remember, all mitochondria will come from pre-existing mitochondria. This is one point. All mitochondria will come from pre-existing mitochondria. So because it is coming from binary fission, you cannot have brand new, you cannot have de novo mitochondria. So all mitochondria will come from pre-existing mitochondria. What this implies, if the pre-existing mitochondria, the initial mitochondria has some defect, if it has some defect, all the other subsequent downstream mitochondria are going to have the same defect. And the second question is, from where is the inheritance occurring? Is it coming from mother? Is it coming from father? Is it coming from both? Can it come from any of them? There is no fixed pattern, it can come from any of them. What is the inheritance? What is the inheritance of the mitochondria? Does it come from mother? Does it come from father? Can it come from both, or it come from any of the sources in the offspring? Means when the ova and the sperm combine to form the zygote. So from where is the mitochondria coming? From where is the mitochondria coming? Quickly tell me the answer. From where is the mitochondria coming when the two combine? Very good. So the answer is mother. Alright. So these are two important points. All mitochondria come from pre-existing mitochondria, and all mitochondria come from mother. All mitochondria come from mother. Because of this, when we look at the inheritance pattern of diseases associated with mitochondria, now coming to the clinical aspect, that is the mitochondrial DNA disorders. The mitochondrial DNA disorders. There we see two important points in inheritance. When we examine the inheritance pattern, we find an affected female transmits disease to all offsprings. An affected female transmits disease to all offsprings, whereas an affected male cannot transmit the disease. An affected male cannot transmit the disease. So when we talk about mitochondrial DNA disorders, these are the two rules which are followed in the inheritance. The two rules which are followed in the inheritance, and based on that, we can have what is known as the pedigree chart. I will not draw the pedigree chart. I know you already know about the pedigree chart, but they will discuss next year in pathology. So I'll not go into the pedigree chart for mitochondrial DNA disorders, but just remember there are certain disorders where the damage is in the mitochondrial DNA, in the 37 genes that we have discussed previously. So there is damage in the mitochondrial DNA, and because of that mitochondrial DNA, we can have certain disorders, and in those disorders, most frequently you will see the lactic acidosis. Why? Because all the proteins that are being formed by the mitochondrial DNA are belong to the electron transport chain. If electron transport chain will not occur, then aerobic metabolism cannot occur and anaerobic metabolism will occur. End product of anaerobic metabolism is the lactic acid, giving rise to the lactic acidosis. So lactic acidosis is the common feature. Along with the very specific inheritance pattern, for the mitochondrial DNA disorders. One of the most common mitochondrial DNA disorders is what is known as MELAS. MELAS stands for mitochondrial myopathy, encephalopathy, lactic acidosis, and stroke-like symptoms. This is one of the commonest mitochondrial DNA disorder. Although the mitochondrial DNA disorders themselves are very rare. By themselves, they are very rare. One of the types of diabetes where the diabetes is associated with deafness is due to the mitochondrial DNA disorder. It is due to the mitochondrial DNA disorder. A condition called MRF, myoclonic epilepsy, with a characteristic histological finding called the ragged red fibers. Is another disorder which is due to the mitochondrial DNA damage. Then we have a condition called Kehr-Sears syndrome. So there are certain specific disorders which are due to the mitochondrial DNA damage. When you encounter these in the pathology section, we have a whole list, about 11 12 disorders are there. But even if you know some names to write in your, maybe if you get a 5-mark question for the mitochondrial DNA, there you can mention some of these names. It will complete the picture, and the complete note will be there when we are talking about the mitochondrial DNA. So let's quickly go over the mitochondrial DNA again. Very quickly, we'll go over the mitochondrial DNA. So starting with the mitochondrial DNA, we said it is double-stranded, but instead of being linear, it is circular. It is quite small, having 16569 base pairs, which includes only 37 genes. Out of these 37 genes, 13 will code for proteins. All the proteins belong to the electron transport chain. 2 of them will code for ribosomal RNA, and the remaining 22 code for the transfer RNA. In the mitochondrial DNA, there are no introns, therefore, the simultaneous transcription and translation can occur. Very few untranslated segments are there in comparison to the nuclear DNA, where barely 1.5% is getting translated. In comparison to the nuclear DNA, the rate of mutation is 5 to 10 times higher. 5 to 10 times higher than the nuclear DNA. And lastly, it shows certain exceptions to the universal genetic code, and we listed those exceptions in the form of a table. Please make note of this table. Lastly, we said that all mitochondria comes from DST mitochondria, and inheritance is exclusively from the mother. So when we talk about the mitochondrial DNA disorders, we notice a peculiar inheritance pattern. An affected female is able to transmit the disease to all the offsprings, whereas an affected male is unable to transmit the disease because he is not transmitting the mitochondria. and because of that you can get a very unique pedigree chart. All the patients having mitochondrial DNA disorder will present with lactic acidosis with the specific inheritance pattern. Some examples of mitochondrial DNA disorders are MELAS, diabetes mellitus with deafness, hemicronic epilepsy with ragged fibers, the Kearns-Sayre syndrome. There are multiple other examples, but I think these 3-4 examples are good enough for you to write the complete notes on the mitochondrial DNA. Okay, so this completes the properties of the DNA. Tomorrow, instead of RNA, I said we'll be doing the practical work. So let me discuss Dr. Sushil and put up a small poll where you can quickly give your opinion whether you want videos also for the practical work or only discussion on the principles and the important biochemistry question, and obviously highlights for the process. We'll go through that. But if you want videos, then I'll have to make the slides with the videos also. We have those videos, that's not a problem, but whether they are required or not, so we'll put up poll for that. Any doubts, any questions that you want to ask from today's class? See, this is a very important topic what we discussed today for your exam. Very frequently you get the question, either the 5-mark question or the 10-mark question on the DNA, the nucleotides, and the next topic that we're going to talk about is the RNA. So it is very important to get a good grasp on the properties of the DNA. A lot of time you get the differentiating question between, we had said, the bDNA and the zDNA, the difference between the mitochondrial DNA and the nuclear DNA. So these points you have to know, and we have discussed those points. Okay, we have one question previously. Abhi had asked, if one of the X chromosome is defective, when it will become bar body, or the other one, how it is decided. I'm sure in your embryology section you have talked about a phenomena called lionization, also known as the heterochromatization. I'm sure all of you have read about it. What is lionization or the heterochromatization? What is lionization or heterochromatization? Anyone can tell me? In between the 32 to 64 cell stage, in between the morula blastula stage, okay, in between the 32 to 64 cell stage, out of all X chromosomes, randomly, see the answer is here, randomly, there is no fixed pattern, randomly activity of one X chromosome is retained. So obviously at the same time all others are inactivated. All others are inactivated. Okay. Okay, so this is the answer to the first question, how it is decided which X chromosome is to be inactivated. There is no particular pattern. There is a random inactivation, or you can say activation of one of the cells. I'll give an example. You see, suppose there are 4 or 5 or maybe 10, maybe 20 people. I don't know how many people are there, and all of you are standing in front of a door. The door to the room is there. Inside that room, there is a paintball gun. Okay, not make it morbid, but there is a paintball gun. So when the door opens, all of you rush inside. Alright, all of you rush inside, but only one person can get hold of that gun. Whoever gets hold of that gun will paint all the other person. So the person who has the gun will be safe. Everyone else will get colored with the paint. Right? So this is what is happening. One of the X chromosome which is able to transcribe the XIST gene will coat all the other X chromosome with that particular RNA. So all the other X chromosome will get inactivated and that particular X chromosome will remain active. This is random. There is no fixed pattern for it. So this is the Lyon's hypothesis, which says that the heterochromatization, that is inactivation of the X chromosome at this stage, is a random. It doesn't have any pattern. That is why if you examine the cells from a female, half of the cells will have the X chromosome inactivated from the father and half of the cells will have the X chromosome inactivated from the mother. The randomization ensures approximately 50-50 inactivation is happening. Okay, that is one. Syllabus for this test, this Sunday grand test. Okay, so heme degradation and today's class that we have done. Heme degradation and today's class that we done. I said it is random. It is random. Okay, DNA repair mechanism, identify which site to be repaired. We have not talked about DNA repair mechanism. Don't worry, once we do the replication, then we will talk about the DNA repair mechanism. DNA ends are complementary. No, DNA ends are not complementary. DNA ends are not complementary, but the sequence in the telomere is self-complementary. It is complementary to itself. It is not complementary to the sequence of the other DNA. It is self-complementary. It will not interact with other DNA because the telomere will fold upon itself. When it is folding upon itself, it will not interact with the other DNA. The term is not complementary, but it is self-complementary. So, like I said, it is not the maternal or the paternal X chromosome which is targeted for inactivation. The inactivation is totally random as per the Lyon's hypothesis. The inactivation is totally random. Any other question that you want me to answer about the syllabus? Again, last week and this week what we have covered, that we cover. Tomorrow's class will not be. Tomorrow's class, the practical work that we are going to discuss will not be part of the Sunday test. The Sunday grand test will not include the discussion that we are going to have tomorrow regarding the practical. Any other question? Any other question? So if there are no other questions, I'll close it here. Alright, I'll close it here for today. So take care, stay safe in this COVID crisis. It is very important for you to take care of yourself, because as you know, exams are just from the corner, and your preparation should not get affected, because if you are not careful at this point, you might get infected and quarantine, etc. That crisis will occur. So please make sure that you're following all the precautions, frequent hand washing, use mask if you're going outside, and all those things. Okay, Abhi is asking if they are not complementary, then how are it pairing? It is not pairing. See, it is getting repaired by the DNA repair mechanism. There is difference between pairing and getting repaired. Repaired means the DNA repair mechanism will add complementary sequences and then join them up. Okay, it will add the complementary sequences and then join them up. So it is a repair. It is not pairing. It is a repair. Okay. I hope it clears it up. That is a repair mechanism which is joining the two strands. They are not joining by their own. Chalo. Take care. Bye bye. Stay safe and good night. Thank you very much for your active participation. It is always interesting to see the questions from your side. Thank you and bye bye.